From Microbe TV, this is TWiM, This Week in Microbiology. This is episode 140, recorded on November 18th, 2016. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. We're coming to you today from the Hamilton Performing Arts Center in Hamilton, Montana. I have a really special episode for you today. We have 3 local high school graduates and 2 teachers, all of whom love science. There's big science in this small town. Of course, that's Rocky Mountain Laboratories. And tonight we're going to see from our participants how a small town and big science can come together. I have a really good friend here in Montana who is an author, David Quammen. Maybe some of you have heard of him. He lives in Bozeman. He writes fabulous science books, and I'll never forget what he told me once. If you want to tell science to people, if you want to tell science stories, write about the people who are doing the science. And that's what we're going to do tonight. And there's no better way to understand science than through the people that are doing it. So let me introduce my guests tonight, right here on my left from the university. I got excited, because it's the University of Montana, where he's a sophomore, Kyle Shiflett. Welcome to TWiM.
Thank you.
From Rocky Mountain Laboratories, Brian Hansen.
Hi.
Also from Rocky Mountain Laboratories, Forrest Jessup. Welcome. And from Hamilton High School, science teacher Marie Antonioli. Welcome. I just love the I's and O's in your name. Racaniello, Antonioli. And finally, former Corvallis High School science teacher. He's now at Rocky Mountain Laboratories, Jim Strebel. Welcome all of you to TWiM.
Hello.
First thing I'd like to do is go through all of you. I'm very sorry. Go through all of you and figure out a little bit of your history, where you're from and where you went to school and so forth. Let's start with you, Kyle. Are you a local guy?
I am, yeah. I've been in Corvallis, Montana since I was 9. Mm-hmm. I moved over here from Georgia. Both of my parents are science teachers.
Wow. And you went to high school locally?
Yeah, Corvallis High School.
And you're currently a sophomore at the University of Montana.
I am.
Right? Mm-hmm. And I think you'd like to get a PhD one day, is that right?
Definitely.
Okay, we'll talk more about that and sort of kinds of things that you have done as well. Brian, what's your story?
So yeah, I'm one of the locals around here, graduated from Hamilton High School in 2004. Rocky Mountain Labs does a middle school outreach program called BRASS, which is Biomedical Research After School Scholars, where we go to some of the local middle schools and do some fun science activities with students. And I got the privilege, because of Miss Antonoli and others, of being in the very first BRASS session, and kind of sparked an interest that I had always had in science, and ended up doing various different internships at the lab through high school and college, and now I'm on as a full-time employee at Rocky Mountain Lab.
All right, we'll talk about what you're doing there in a bit. Forrest, tell us your story.
Well, I'm born and raised here in the Valley, and went to Corvallis High School. It's where my long journey in science began. And
not so long.
Yeah. Well, my life's I'm not that old yet.
That's right.
So for me, it seems like a while. And I got my undergraduate at the University of Utah, and did my graduate school actually back up at the University of Montana. Knew some great people up there, and now at Rocky Mountain Lab. So unlike some of the others here, I actually never really did any internship or anything at Rocky Mountain Labs. This is my first go at it, though I've always been familiar with it and have benefited from it as well. So
you don't need to go through the same channels that these individuals have to get there.
No.
There are multiple paths. Okay, very good. Marie, tell us, where are you from?
I'm from Butte, just down the road, 3 hours. And I've been teaching here at Hamilton for 25 plus years, both in the middle school and the high school. I've taught a variety of different science classes and had the wonderful opportunity of doing science fair for a long time and science symposium. And lately I've been teaching mostly health enhancement, and have been partnering a lot with different scientists at Rocky Mountain Lab, different people there, I've had wonderful relationship with the lab that I'd love to talk to you about.
Okay, we'll explore that. Jim, what's your story?
It's long and sordid, but I guess
Oh, I'd like to hear the sordid part.
I came to Corvallis in 1992 from University of Michigan. I lived all over the U.S. growing up, but my parents were both from Montana. My mom's family came across the border from Canada and homesteaded here, and my dad went to high school in Deer Lodge, so I had connections to Montana, and I kind of came back here after college, but I taught at Corvallis for 14 years, and then was lucky to find a place at a Rocky Mountain Lab, and I think for the 24 or 5 years that I've been here, I've always sort of been in the science community and kind of in the loop of Rocky Mountain Lab, and it's always been sort of near and dear to me.
It's an interesting series of events, so we'll get back to you and explore that and see what you did. So Kyle, at the moment, you're a sophomore. At what point, so you had some experience at Rocky Mountain Laboratories here, that maybe they got you interested in science. Why don't you tell us a little bit about those?
Well, it was kind of, I'd always kind of been known I kind of wanted to do something sciencey.
How did you know that? Was it an outdoorsy type thing? A lot of people start that way.
Partially that. I grew up a lot in Yellowstone. Both my parents being science teachers is kind of a family thing as well. But yeah, just being close to the lab, and my sister having worked at the lab a little bit before me was really kind of my kind of entrance into it, kind of knowing about the lab and wanting to work there and wanting to get more into science. And that was kind of the lab was kind of my gateway to do that.
So you spent some time working at Rocky Mountain in the summers, right?
I did, yes, 3 summers.
Tell us about that.
Well, kind of how I was kind of got in was I applied. So the lab does a program where they hire high schoolers kind of to clean glassware and keep upkeep on the lab.
They pay you?
Yes, they do.
Great.
And I kind of applied to do that. My sister had done that. I thought it'd be great to just work around people who were actually doing science, and something I might want to get into in the future. And in my application, I mentioned how much I love science and how I might want to do that in the future. And they responded with, well, hey, if you love science, how would you actually like to do some science here? How would you actually like to have an internship program over the summer? And of course, I was elated that I would get that kind of opportunity. And so for 3 summers, I came back and did the internship program.
Can you tell us a little bit about what kind of work you did in the labs? I think you had several different experiences, right?
I did, yes. I worked in the laboratory of virology, and the first 2 summers I worked with arenaviruses and kind of some reverse genetics systems for those, Hunin virus especially, kind of a lot of cloning and looking at kind of reverse genetics assays and antibody responses. And then around that time, the mentor I had had up to that point, Thomas Honin, moved away, and Andrea, who's in the audience, actually welcomed me to do another internship under her, and she works a lot with vaccines and Ebola virus. So I got to work on a Ebola virus vaccine project, which was great. It was right around after the time of the Ebola outbreak in West Africa, and it was just like, it's very cool, a great opportunity to be able to work with that in high school.
I can't imagine how many high school students would have that opportunity.
Yeah,
right?
it's great. It was
pretty
honestly
unique. Probably only here it could happen, actually.
Yeah, really.
Not many other places to do that. So you got to love viruses, maybe?
I got to learn to love to work with them, especially. They're very interesting. It's the way they can just change and evolve to adapt to anything is really amazing to me. And studying that was definitely kind of solidified, like, hey, I want to do this. I want to do research.
Yeah, there's nothing like a good experience. It's really neat how you say you started washing glassware. And on our podcast, people often say, how can I get into a career in science? We often tell them, go volunteer in a lab and wash glassware and show that you're earnest and interested. And then they'll say, hey, you want to do some experiments? You got paid for it, which is pretty good.
Yeah, it's very good, yeah.
So, as you know, I'm a virologist. And did you know, for example, that all of us are full of viruses at this very moment?
I did.
They're coursing through us, but we're fine. Isn't that amazing? That's got to be something you want to pursue even more. Well, we'll talk a little bit about some of the things you've done in a moment. But Brian, you are working at RML, right?
Yes.
You're working
yes.
in the electron microscopy group. Tell us a little bit about what you're doing.
Yeah, so I help with a lot of the day-to-day microscopy work that we do, looking at different viruses and bacteria and what happens when they get into a cell, or what happens when you bind an antibody to the surface or things like that. And what I've been doing a lot lately has been a lot of computational biology. So we have a lot of high-end microscopes that can output a great number of images, and then we can take those images and isolate particles from them and then start averaging those particles together and generate very high-resolution 3D models of a virus or a protein complex within a cell. And then we can do some fun analysis and identify interesting regions for vaccine candidates and viruses and things like that.
So have you actually prepared specimens, put them on grids, and put them in the microscope and viewed them and all that?
Yep. Yeah. So when I first started in the group, I was tasked with a project of looking at specimens frozen in a scanning electron microscope, which looks at surface details mostly, but if you freeze a sample, you can then fracture it open and then look at it under frozen liquid nitrogen conditions and start to get an idea of what's happening inside the cell, even with a scanning scope. So you can see large vacuoles filled with bacteria inside of cells and some pretty fun stuff.
Don't you find it amazing that you can take all this stuff, the samples, in this huge machine, you turn on the vacuum, and then you can see things that are, isn't that amazing?
Yeah, it's pretty incredible, what we get to see. And, you have huge telescopes that look out into the stars, and we get to look at it from the other end. We have large microscopes that let us see very small things. One of our microscopes, one of the resolution specs on it, we can actually see gold atoms inside the microscope. And to actually look at the computer monitor that's showing the camera image on it and actually be able to go, that's a gold atom, is a pretty unique, interesting thing.
So I understand that you got into this EM via the BRASS program. Is that correct?
Yeah, so I did BRASS and really got interested in that. And then in high school, I worked in a lab that's part of the Laboratory of Persistent Viral Diseases and got to do a lot of really cool work with that group. My mentor at the time, they would give me little snippets of a mouse tail, and I got to extract the DNA out of the mouse tail and then do some PCR and other tests on it and figure out the genes that are in the mouse that they're looking to knock out and which mice they needed to breed to get the knockout program, and got to do some cloning experiments. And by the end, you don't really realize what you're doing until you get to college and you're like, oh, those are senior and graduate level experiments that I was just performing as a high school student.
It's great. I tell you, I didn't do any of that as a high school student. So that's really good. Now, this BRASS program, it stands for Biomedical Research After School Scholars. Is something you put together, Marie?
Well, I didn't put together, but when I was teaching 8th grade integrated science, I've been talking to different folks at the lab and saying that we were noticing some different surveys that said that science interest was dropping off at the middle school, especially among girls. And so we bantered back and forth about maybe some way we could address that. And we started off as just having a few guest speakers come and talk at the middle school. But then Anita found a program that they'd been doing at Bethesda that had using postdocs and so forth from the lab there getting into the middle school. And so it's been 18 years since the BRASS program started. And I can't remember, let's see if I can see it here, how many, over 1,000 kids, 650 postdocs have been a part of it, and the kids elect to do it. And so what's powerful about that, you put the learning back in their driving then. They know what they want to do. And it also helps because it's not a gifted and talented program. It's any kid can get into it. So some of these kids that might be a little lackadaisical at school, you get them into that program, and if you could watch what some of these folks do, it's just absolutely amazing. And these kids just get lit up. I mean, it's just phenomenal to see it.
So part of the program is visiting RML. Is that right?
They have done that in the past. They alter it all the time. They have, most of the time they have after school stuff where the kids come and do active science with an actual mentor postdoc.
Okay, so Brian, that's how you got interested in RML through the BRASS program.
Yeah,
right?
and I've had the privilege now since coming back of actually going and instructing some of the courses, and like we were saying, it's really incredible to see the interest that these kids have. We've done sheep eyeball dissections and sheep brain dissections and different things, and eye tests. Looking at how your eyes respond and brain injury and epidemiology, how vaccines can help stop spreads of viruses and things like that. And it's really cool to see how quickly they can grasp it and form meaningful conclusions in the experiments we have them do.
That's the best way you can perpetuate science, to go back to school not too long after you leave and say, hey, look what I did, right? And you could do this too. Because they may not believe a teacher, right? But they'll certainly believe a graduate.
Absolutely, that's right.
So you mentioned that you're doing some reconstruction, EM reconstruction. Do you do viruses, for example? Or?
Yeah, we've done some poliovirus modeling, norovirus, hepatitis B. I think those are most of the viruses that we've mainly focused on. We've done some work with influenza and how it binds to a plasma membrane and forms a pore and some fun stuff like that.
So you were taught to do all this at the lab, I presume, right?
Yeah, it's been a lot of on-the-job training and going to different workshops and classes and having really great relationships with the people that develop the software that kind of does most of the work, and being able to go back and forth with them when things don't work or you have a problematic sample. They can give you a lot of tips and tricks on what you can do.
Did you have computational skills before this that allow you to easily move into it?
I've always liked science and computers, and never really thought of putting them together too often, And then as I started in the EM group after college, we were getting into a lot of what's called tomography, which is basically taking our sections that we use for regular transmission electron microscopy and performing an MRI on them. And I really saw a cool area of mixing science and computers together and doing a lot of fun stuff.
So you can build like a MRI, you make a three-dimensional image. You can do that with biological samples that are really small.
Mm-hmm. Yeah, it's computationally in the software, it's all exactly the same as what hospitals use for MRIs. We can take part of a cell and build the volume and be able to see, what effect an infection is having on the internal mechanisms of the cell. How is the bacteria or virus evading the immune response, or what's it doing inside the cell that's letting it replicate?
Great. All right, Forrest, you're at RML also, but you did some work in high school as well.
I did.
Tell us how you got to do that and what you did.
Well,
Everybody's laughing
put it
here.
crudely,
There must be a story.
Started with crap. That's how my journey into science started. Was in high school. Was isolating DNA from Rocky Mountain goat scat.
So really crap, right?
Yeah, really. Yeah, and this is stuff that. So I came a little bit late on the scene. Jim and the science program at Corvallis, they had a science program called Classroom Without Walls, and I actually wasn't a part of that, but they had gone hiking up into the wilderness and collected a bunch of goat scat and brought it back. And
how'd they know it was goat?
you can't identify. There's like nice little lumpy pattern there. So.
And you could watch it fall too.
Yes, you could.
And then you know.
Yeah, well, that's true.
Yeah, they had a goat that followed them that they could probably just feed off of the whole time, just kind of wait.
There's a certain population of goats that show up every morning outside your tent. You unzip the tent, and there they are looking at you. And samples are there.
Okay.
Yeah.
I can understand that would be a goat, but you also collected scat that's already drop, but you can identify it.
Yes.
Has a certain morphological quality, right?
Yes. And well, a lot of that was brought back to the lab and was literally frozen down in little 1.5 mil epi tubes. And I came in late. I was interested in doing science. I hadn't really committed to doing science. I just thought it was kind of fun to do, and it turned into a lot more. The goal of the project was to one, get it to work, but two, we were trying to see a genetic diversity between populations. So we were looking at microsatellites, doing microsatellite genotyping, and in a lot of ways, we didn't really get that far, but I was able to learn through the process how to isolate DNA and how to do PCR. It took me about 6 months to get a band on a gel that I'd poured on my own. And I worked on it on my own. I didn't ask a lot of people how to do it. I just kind of had to figure it out over and over again until I got it. And then from there, we kind of started getting it going, and we actually got a little bit of data, enough to submit an abstract or two here and there. We went to the state science fairs.
But before you get to that, the day he got the band on the gel, I mean, he was screaming in the back room, "I got the band! I got a band!" He was so excited.
I don't remember that.
It was a lot. You don't. It was a lot of hard work.
It was.
And he did at lunchtime. He came in after school when the other kids were doing other things. He was isolating DNA from goat scat.
Yeah, and this is where one of my indirect connections with Rocky Mountain Lab came in, is they actually gave us a bunch of surplus equipment. An old spectrophotometer so I could look at the quality of my DNA that I had actually isolated. PCR rigs, I still have to pour my own gels, couldn't buy those. And there's some other things too, tubes, tips, pipettes, centrifuges. I was able to learn how to use all those things, and then we went to the science fairs, and I did fairly well at the Butte Science Fair. They had the regional one in Butte. We actually got a first place in that one. And if you won a first place in the regional science fair, you were able to go to the international science fair. I also went to the state fair, did okay at the state fair. And then I went to Salt Lake City as well for a symposium, and I cannot remember the name.
Science and Humanities.
Science and Humanities
Yeah,
Symposium.
Western
Yeah.
science.
And I got a third place in that one with this project, and was able to go down to Albuquerque. So we kind of joke about it now, but this was also Jim's last year that he taught at Corvallis, and the whole last half of the year he didn't actually teach much. He just traveled with me to different places.
True.
And we did fairly well. So a lot of this paid off, and I'm saying we here because there was actually two other students that helped out with this project. They're not here. They're off in school and other places. Jeremy Reynoso and Alyssa Schlecht, and they helped with this project as well. And so I did a lot of the hardcore science stuff, but they did the picking up the goat poo and bringing it back and mapping it out on maps where they got it and whatnot, and helping us write a paper for it. And we did well at the International Science Fair. We got a second place, and that came with award money and things like that. And a lot of important skill sets that I was able to go into college. And I think that's one of the most important things that came out of this was we had a visiting speaker came down from University of Montana, Dr. Christopher Malaccio. One of my friends, I've known him for a long time now, worked with him for a long time, and he's an immunologist. He came down and did visiting speaking, and I asked if I could job shadow him. Through this opportunity, because I've had these skills, that job shadow opportunity actually turned into a job, a summer job. I started 2 weeks out of high school working in a lab, and I wasn't washing glassware. I was actually on a project. Not that there's anything that's very good to do. I was very fortunate to come in, and I was handed a project right off the bat that turned into about 2 papers. I wasn't first author, but 2 papers within about a year. So it was great, all this kind of stemming from the experience that I got.
So the project you're talking about, the goat scat project, was that done mostly at high school?
Oh, all of that.
All of that was done at the high school. You didn't do any, at the time, no RML involvement, right?
Nothing was done at RML other than
that came later.
Equipment. That came,
So I understand the prize for this second place, you got a planet named after yourself, right?
Yeah, I don't know. You brought the information.
Yeah, we printed out the sheet because to me this is kind of a culminating event that, you know, a part of my life and his life where they came together, and the planet, it's in the outer main belt of asteroids between Mars and Jupiter. And it's about 6 miles in diameter. It's really an asteroid.
So it's called Forest now?
It's called Jessup.
Jessup. But I thought it's named after you. But your last name, sorry.
Oh, there's a lot of them. I guess it could be.
Yeah, okay.
It could be a lot.
So
but it is supposed to be me.
Do you have a photo of this in your room?
No, but they gave us a nice little, almost looks like a diploma with all the information in it. You can
That's
pull
cool.
out and things.
I want to know when you're going to go there.
I asked for mining rights, but they, I don't know.
So then you, oh, by the way, this goat study, you said there were no conclusions that you could make from it?
Not really. We had some goats that we identified as being heterozygous for certain microsatellites, and others that we didn't, but that's about as far as we got, as we were able to start seeing the diversity within the goats. I got all the protocols to work, and that's kind of where it ended, and handed it off to other students that I know. Some have actually kept parts of it going here and there throughout the years, but I haven't kept up on how far it's actually come.
Yeah, it's kind of morphed into some other projects.
I'm sure it has.
We'll hear from you about it maybe. So you got a BS at the University of Utah?
Yeah, I started off at the University of Montana. I did 2 years in the working with Chris Malaccio. He was actually a senior postdoc at that time. So really, I was working for the head of the tox department up there, who's Dr. Andre Holian. But I was working on Chris's projects, and he was doing a lot of macrophage biology. He just loves that stuff, and I do too. And a lot of that was focused on particle work, so it was a lot of particle-induced lung injury and things like that. And 2 years in, I kind of started to think I hadn't quite fully committed to science. I knew I was going that direction, but I was thinking of med school, actually. And I did, partway through, I transferred down to the University of Utah, thinking that would give me a better chance of getting into med school. And I worked for 3 years for one of the best people I know, and that's John Hoytel, Dr. John Hoytel. He's the chair of internal medicine, or a former chair now. He's just stepped down earlier this year. And working in his lab, and I got to work on a wide array of diseases. Just being in internal medicine, he would show up and say, well, here's cystic fibrosis, sputum,
It's better than goat
do
scat,
something
right?
with it, kind of. We had ideas, but that's wasn't just do something with it, but you get the idea. And different models of injury that we were doing at the very end. I was doing cecal ligation and puncture, which is, for those that don't know, it's a way to induce sepsis, and you can get your intestines can get perforated, and you can leak bacteria into your body and then get sepsis that way. And so we were doing some models with that near the end. I was exposed to a broad range of areas in science, as well as what MDs did, because this is a medical school. Most of the people I worked with were MDs. And nothing against MDs at all. They do great things, but a lot of them didn't have time for family as well. It's a very intensive job. And I was getting married, and I wanted kids. And really enjoyed doing research. Not just that, I really did enjoy doing the research side of it. And so I decided to do research, and I knew the program back up at the University of Montana, the toxicology program, is a great program. Andre Holian is fantastic, a fantastic mentor. And so I applied to that, and I had some experience, and I got in as a graduate student, and here I am. Doesn't explain how I came to RML, but
we'll get to that.
Yeah.
We'll come back to you. Absolutely. I want to talk about RML and what you're doing there, but let's go to Marie and talk a little bit about the things you do in high school to get kids interested in science. We talked about the BRASS program. One of the things I noticed here, you raised some money to teach a biotechnology class.
Right. So, several years ago, we thought, here we have two wonderful labs in our small community, Rocky Mountain Lab, and then it was Carix, I believe, at the time, and now it's GSK, GlaxoSmithKline. And we thought, wow, there's all this science here, but then we don't have the ability to really teach the kids and for them to kind of at least get an idea of what that sort of science is. So it was mostly my husband, who is the biology teacher, who's sitting over there. Thank you for coming. But we wrote a grant, and some very generous local people, the Ryans, gave us money for updated computers, PCR, just everything really that we wanted. Wonderful electronic microscopes. And so we started that quite a while ago, and it's still a very, very popular class. And I just think it's an essential part of our curriculum, given where we live.
So this is not just a lecture, it's a hands-on as well. They do
It's
things.
very hands-on, lots and lots of wonderful labs. I taught it for a little while, and my husband's been teaching it for the last several years. He does a great job. And it is an elective class. So the kids who are there are there because they really love science and they really want to learn about it. They're not all the top 1%. We get all kinds of kids, and they learn a whole bunch. So it's great.
So in all the conversations I've had with people who have gone in science, I always ask them what was the catalyst, and there are two or three main ones. One is, interestingly, being outdoors. A lot of people as kids are outdoors, and they get to like nature, and that leads to science. But another main one is having a great teacher in high school. So that was me. I had a fabulous biology teacher, but she didn't do any of these things. She just talked to us. And, I listened to the things that she said. She used to work in a lab. She didn't have a biotechnology course. She had no equipment. In fact, when I went to high school, there was no PCR, there were no DNA gels. It was pretty primitive.
Yeah.
Yet, she would talk, her eyes would light up.
Right.
And that was really what got me excited.
And you've hit on a very key thing there, is that when we try to get kids fired up about science, having guest speakers, having the BRASS program, being able to work with mentors, it's that passion that really the kids that's what they relate to right off the bat. When someone loves what they're doing and they are able to express that, right away the kids pick up on that and appreciate that so much. And then the second thing that the kids really appreciate is when whoever it might be, whether it be the teacher or a mentor or a guest speaker, but they're observant and they kind of meet the kids where they're at. And they kind of get to know the kids a bit, and then wherever those kids are at, then they go from there. And sometimes, there was just an episode of a television program where these scientists were being guest speakers, and they were talking way over the heads of the kids in the audience who were checking out. But that doesn't happen. The people who come and speak to the kids, they really make an effort to try to tap into what those kids are interested in. And then, like I said, just being able to express their own passion for science does more than you can imagine.
I think at any level, teaching with passion is the key. Not just high school, but college as well. And it's really important. Students respond. So we were talking before about how it's really unique to have a high school person work on Ebola, and you did. I think that's a key where a town like this with a big lab can make an impact. because not every town in the U.S. has a lab that's working on Ebola. So you did something with Marshall that relates to public information about that.
Right. So we have a health careers class, and it's considered a career and technical education class where the kids are allowed to kind of explore lots of different health careers. But one of my visions for that was to, again, try to make the learning more active and put it back in the kids' court so they could kind of decide what they wanted to do. So 2 years ago, during the big Ebola outbreak in West Africa, the class, this health careers class, decided they wanted to learn more about Ebola. So the first thing was, okay, let's ask someone to come over. And so I just emailed Dr. Bloom and said, hey, could someone come over and talk about Ebola to my health careers? He said, sure, I'll come over. And then things started to go from there. And with his help, the class took over the learning and decided, hey, they wanted to not just learn about what the disease was, but they wanted to know what our local labs, both of our local labs, were doing to address it. And then they wanted to figure out what would be a fun way to teach other kids about it. So they devised videos to, pop songs, and they had interactive things with mortality rate. They had, little lifesavers in a bag they give to people talking about the quest for a vaccine. But they're the ones that developed all of that, and they absolutely loved it, of course. We had the whole student body of Hamilton High that came and watched that, and then we got calls from Corvallis High School, and they said, hey, we'd kind of like to hear about that. And so then they came over, and then Victor High School said, oh, that sounds pretty cool. How about coming over to Victor? So we did. And those kids will always remember that, partly because it was such a topical thing at the time. It was something that they wanted to teach their classmates about it, the safety and also the research of it. But they also will remember it because they got to choose how they wanted to discover the learning on their own.
I think you reached out to the right person, but not every high school teacher knows who to reach out to. And so I would say, to any high school teachers listening, and we do have quite a few, just email. That's the beauty of email. You don't have to call anyone now. You can email, find a name at the local university, and just keep trying. Someone will respond and help you put together something like you did.
Right, that's great advice.
It's easy to do, because every university has a Marshall Bloom.
Me too.
Tell us a little bit about your three daughters. You said they've had experiences at RML that were interesting.
Right, so I have three lovely daughters. The oldest is 21, the next one's 18, and the youngest is sitting over there. She's 16. And they've all three been very lucky because they applied and were granted internships, student internships at Rocky Mountain Lab, all three in different labs. And so maybe I'll start with the youngest. I think she went into this just this fall, not really knowing what she wants to do. And then she's working in genomics. And just last week, she did a presentation in the health careers class, and she wants to go into anthropology and do genomics now because there's someone in the lab she's working with who has just totally lit her on fire and has really shown her that this would be a wonderful application of some of the things that she does there. And she's learning all kinds of wonderful techniques. And I can't really speak to that because I don't really know all the things that she's doing or understand them, but she absolutely loves her job and is hoping to continue there.
So
let me just talk about my next daughter because she, I'll talk about all of them, but my middle daughter just graduated, and so she was able to work with Brian, which was amazing, and she did a lot of work with microscopy, but she's an artist, and through her internship, she was shown that she didn't have to necessarily choose between being a scientist or an artist, that there's all kinds of avenues where she could blend those two passions. And she was the kind of kid who, if she had the choice going out with her friends or working longer at the lab, she'd much prefer working longer at the lab. It's continued to be her passion. She's at college and she's continuing that. And then the oldest, well, she's decided history's her thing, which was good because it saved us a bunch of money that, you know, she kind of found out early that, yeah, science, yeah, it was kind of fun, and she liked her job, and the people were so kind and welcoming and supported her so much, got to know her as a human being. But yeah, no, history was where her calling was.
The 2 out of 3 isn't bad, right?
No, I'm pretty happy. Yeah.
So you've succeeded. That's very
Yes,
good.
yes.
I mean, I have 3 kids, and two of them are computer science majors. Does that count as science?
I think so.
Yeah, I think it does. Okay. And my daughter is an English major. So I'm like you.
Right.
But the initial, them going to RML, was that, did you have to convince them, or was it easy?
Well, I think that all of them were very intrigued with the fact that Rocky Mountain Lab is very flexible in letting kids work when they can. So if they have, you know, sports in a particular time of the year, they're pretty good about working around that schedule. Or if we have Honor Pass and Senior Release here that enable kids to actually go to a job during the day. And so that was very intriguing. And you know, the pay was good. You know, it's $10 an hour, so that's better than McDonald's. And so I think because also they have a science interest, it was something that's a competitive thing that you have to apply for. But they were all very happy to secure those jobs.
Jim, let's talk a little bit about what you've done. You came here, you said, because it's a small place, you could do things, make things happen in this high school.
Yeah, I think I didn't know what I was getting into when I came here. It was kind of by chance, but when I first started at that school at Corvallis, the principal was progressive and interested in letting the teachers, you know, explore. And this was before No Child Left Behind. So there was a little more freedom, you know, not to stick to the test, which was great, I thought. And we kind of focused on what was called authentic outcome learning, where, you know, we'd start project-based kind of things, and kids would have to not just get a grade at the end, but present something or do something that's real in the world. So science fair is, you know, that's real. There's peer review at the end. But we had these projects, like we monitored streams for about 10 years, looking at how the effect of fires on silt and how that would affect benthic macroinvertebrate populations. And then the kids would go give presentations to the Forest Service or to the Trout Unlimited. So we wanted to, you know, kind of do things like that that had real outcomes, not just grades. And that's where I kind of got involved with Rocky Mountain Lab and Marshall. Marie's husband, Tom, and a few other teachers, we started the Bitterroot Science Alliance, which was a group of teachers, scientists from the Forest Service, from RML, from GSK. In those days, it was Rebe Immunochem, I think, before it became CRIXA, then GSK. But the intent of that group was to get good science going. You know, we have these resources, let's get them out to the schools. And that's what happened.
Again, this is an amazing opportunity you have here, and it's great that all of you took advantage of it, bring the students to it and it to the students. I mean, it's this Bitterroot Science, what is it called? Bitterroot Science Alliance. I think this is terrific. So what kind of things did they do as part of this?
Of the Science Alliance?
Yeah.
It was really just sort of a forum for all the scientists and teachers to get together and make connections. We ran an annual science fair every year in Corvallis, and I would bet there's not a small town in the country that had 30 PhDs as their judges, you know, at a high school science fair. And so these kids were scared. They were shaking in their shoes, and they said, "I think these are real scientists, Mr. Streevel." And I said, "They're real scientists, yes." So that was one thing that happened. But we tried to find connection anytime we could, you know. I think one day, a story I remember, the Howard Hughes Medical Institute in Bethesda puts out these publications, and I used to order them via mail before the internet, and they'd send me a big box of books, and we'd read them in class because they were about kind of the new things that were being discovered. And in there, one time we were reading about hematopoietic stem cells, you know, how blood cells are made. And it said this guy's name, Jerry Spangerud. And I said, oh, Jerry, he's at the Rocky Mountain Lab. Or actually, a kid found it. And I said, oh, yeah, he's at Rocky Mountain Lab. And so we called Jerry and went over to Rocky Mountain Lab. And I think they had just gotten their first cell sorter there. Does that sound about right? Mid-90s. And he showed us a cell sorter, and that inspired so many kids. They couldn't believe that they could see single cells flying through the air. You know, it was pretty neat.
It's cool.
Yeah.
So then this seems to have started to insinuate you at RML. You started doing summer work there. Tell us how you ended or made the transition from a high school teacher to a scientist at RML.
It was a hard transition because I loved teaching, but it was a good move career-wise. But I got 3 fellowships, 3 summers in a row to work in Kim Hassenkrug's lab and study friend virus. And these grants were from the M.J. Murdoch Charitable Trust in Portland, and they have a wonderful program there that gives money to teachers to go work in labs in the summer and learn how to do science. And so I got that grant for 3 years and worked in Kim's lab and worked on another degree at Montana State at the same time. And Kim Hassenkrug at the lab was one of my committee members. And so some of my coursework actually was at the lab too. It was kind of a neat program to go between MSU and Rocky Mountain Lab. But that kind of, you know, I worked 3 more years after that at the high school, and then in 2006 I started working at the lab.
So when you did these summer stints, did you have in your mind eventually that you would want to go to the lab and leave teaching?
No.
No?
Not yet. No, I didn't. I mean, that's where I kind of learned the microsatellite genotyping.
Yeah.
We were studying friend virus, and there was a gene that was gave resistance to friend virus, and it was unknown what the gene was, and so we were trying to find out really what this gene was, and I learned those techniques, brought them back to the lab, or back to the high school, and that's where we started this mountain goat project that Forrest eventually took over. So I didn't know then, because I really liked this back and forth that we had with the Rocky Mountain Lab, you know.
So what was the point that you did know that you wanted to go there and leave?
It's hard to put my finger on that, but I think it was talking with Bruce Cheeseborough, who is my current boss. I had known him for a while, and he had an opening in his lab, and I applied for that job. I think a lot of it was monetary based. I mean, it was unfortunately, you know, teaching in Montana is, you know, not a lucrative job. And
I don't think it is anywhere.
yeah, and, you know, to get ahead a little bit. But I think still it's a go-between for me. I still teach at the college here and think that, you know, it's a good way to bring science to the schools.
So what are you doing at the lab? What project are you working on?
I work on prions, so infectious proteins, and I think a lot of people are familiar with prions these days. There's no genetic material, but we work on how prions cause disease, so the pathogenesis of prions. And I mean, you want more details on?
Sure.
Yeah. So I study. I'll give you one project that's probably the most interesting to me. The normal function of the prion protein involves the clearance in the synapse. At least some people think that, and so I've been working on that for a few years. How the prion protein is a extracellular protein, and there's some evidence that it helps in uptake of glutamate from the synapse. So after neurons fire, there's all this glutamate left in there in the synapse, and they need to clean out the synapse so the signal doesn't keep firing. And so astrocytes do that, and the prion protein, some people think has a role in that. And we published a paper that said it did, and for the past 4 years I've been working on that, repeating that work, but in the end we think that the prion protein might not have a role. in all of this.
It's science.
So that's science, and that's
is.
what I loved about teaching too. Was when
Yeah.
we did these, science fair projects, you really learn the methods of science, and you learn that it's not so easy to figure something out.
No, not at all. I mean, but we need to tell people that because a lot of people think it's easy to do that. But we have to make sure that it's clear that it's a journey.
Right.
And but in the end, it's a valuable journey. It's worth doing. Do you miss teaching high school?
Yeah.
Maybe you always wanted to be a scientist.
Yeah.
Okay, I'll come back to you, Kyle. So you're a sophomore in university. You're majoring in microbiology.
I am.
You're doing any lab work?
Not right now. I'm kind of transitioning. I'm trying to get into a lab up at the university right now.
How are you doing that? What's the plan?
Right now, I'm just trying to talk to some professors that I have.
You gonna wash dishes for them?
If I have to, yeah.
What kind of labs are you interested in?
Definitely some bacteriology labs. I've been doing a lot of stuff with viruses, and I think it'd be interesting to see the other side.
You'll go back to viruses. Bacteria, the dark side. You want to go back. So you would like to do some lab work next couple of years, right?
Definitely, yes.
What's your ultimate goal? Do you want to go back to graduate school and get go to graduate school and get a PhD?
Definitely, yes.
Yeah.
And do some professor time too, because I think I'll really enjoy teaching. I like kind of like teaching other people things.
So you're absolutely sure about this?
Yes. I don't know what I want to get a PhD in, but
Yeah.
I know I want to get one. just kind of flipped on how people normally.
And so you'd like to be a professor one day and teach and do research?
I would really like that, yeah.
Mm-hmm. And maybe microbiology related field will do that?
Yeah.
Yeah. Well, I already asked you how you got interested in a career in science. So here's another question for you. What's the best science experience you ever had?
Whoa, that's a tough one. There are a couple moments, kind of, especially working with the kind of Ebola antibody assays that were like, kind of blew my mind that I'm actually working with some serum samples and just looking at a vaccine that someone hasn't really looked at before and seeing if it protects. But there was one moment that I wasn't directly involved in, but I had done some things for an Ebola virus mini genome assay with Thomas and my mentor, and he was testing something for someone else. It was like an MXB protein of some kind looking at an Ebola virus. And he had just finished the assay, looked at the results, and he turns around with it and he has it in his hand and he says, this protein directly inhibits kind of slows down the transcription and translation of Ebola virus in human cell lines. And right now we're the only two people in the entire world that know that. And that definitely blew my mind. That was, as a sophomore in high school, that was kind of, I had to hold on to my chair,
I think that's good. Yeah, I've heard that one before. But eventually, many more people find out, right?
Oh, definitely. That's the goal, at least.
Yeah, of course. You want
Can't really keep it to yourself. That doesn't help anyone.
So if you wanted to, so you love science, you want to do it. If you wanted to convince somebody else to do science, what would you tell them?
Just try to convince them, or just tell them?
Yeah, you say you're in a high school class and you're talking. You've come to one of your old teachers' class, and you want to get them excited about science and maybe get some of them to go into careers. What would you tell them?
I think just about everyone has some kind of curiosity. Everyone's curiosity points in a different direction, but everyone has some sort of curiosity about certain things. Some people it's directly related to science, but other people, can't really see how science would really help them understand about animals or trees, or they like hiking, but they don't really understand how science can kind of play into that. But really, if you're curious about something, generally a science is a way to get more answers, or to be able to learn more about it. Science can be more than just, you don't do science, science guides you. You kind of use science to get an answer. So I'd kind of say, if you're curious about something, look at scientific ways of going about things. Right. That'll definitely get you some answers. So
Brian, do you have an exciting moment in science that you could tell us?
My first one was very similar to Forrest. I was working in high school at the lab, and was working on cloning a bit into HIV envelope, and HIV being a retrovirus, the envelope really likes to seal up and twist and open up again. And I had spent an entire summer, I had run 200-300 PCR experiments and gels, and it was the last day before I had to leave for college, ran the gel, and I finally got the clone to work. The insert happened. I had the band I wanted, and I was running through the lab. And
Yeah, that was a good time.
More recently, you know, the very first time we were working on one of these projects where we were trying to build a virus model, and finally getting all the parameters right and actually having a model of a virus that you can spin around and look at and actually start to see where things are happening on the surface of a virus and possible effects of that is pretty cool. And I think for all of us, all of our stories kind of have a similar thing where the lab is a great resource to have, a great place to go learn, but it starts with the teachers in the Valley and really having excitement about science and reaching out to the lab, because the lab being here in and of itself isn't really enough, for a lot of these great resources to be utilized. You need teachers in the schools reaching out to the lab and getting the students excited so that they want to go to the lab and do science and try out internships, because a lot of people, they think of a scientist, they see the mad scientist guy, with a Bunsen burner and a beaker and crazy hair and funky
it's true.
colors. But there's so much more to it. There's some of the stuff that I do with computer science and genomic sequencing, and our visual medical arts group, you have to take that wet bench stuff and you need to find a way to disseminate to people that don't always know science. You have to find a way to get them involved and make it understandable to them. And that's what I still find a lot of fun about science.
So if you were going to try and convince a high school class to go into science, what would you tell them?
A lot of that. You got to get out and try it, do internships. A lot of times you have in your head what you think someone does, but until you actually see them do it, you don't know. And just because you think of science and the beakers and that and cleaning, there's a lot of other stuff that science can do too. There's artists that make great depictions of what happens when a virus enters a cell. Some beautiful cartoons and colorizations that go on the covers of journals and get used in news stories all around the world. And, it's still science.
Yeah, the best way to teach people is to give them a picture sometimes, right? And if you can make beautiful bacteria and microbes and viruses, that's a great way to do it. So you're working now in the EM lab doing computational work, and what's in your future? Do you have any idea?
I'd like to go on and hopefully get a master's in computational biology, just so, I feel like I have a better grasp of a lot of the math behind some of the stuff we're doing. But I love what I'm doing now, and I love being able to go out to the middle schools and teach, and go into the high schools and talk to the students and help, help impact the next generation of scientists.
So you would get a master's and continue to work at RML?
Yeah.
So you're happy here as far as you can see?
Yeah, for the
So it's
foreseeable
challenging,
future.
right?
Yeah.
Very good. So Forrest, we didn't hear what you're doing. You're working in Katie Bozio's lab at RML.
That's correct.
Who I know very well. And
Katie shares the same fondness for macrophages that I do.
You have an inordinate fondness for macrophages?
I do.
So what do you do with macrophages?
So we're very interested in, so we work on the bacterium Francisella tularensis, highly pathogenic bacteria, of course, as most agents are here at RML. And this is a very strange bacteria. And one of the things that it does, it really prefers macrophages and the lungs, things that I had worked on in the past. So I actually didn't have a huge background in micro. I'd taken classes, I'd had experiments that I had done, but I had a lot of experience with macrophages and lung biology and inflammation and immunology, et cetera. And one of the things that really interested me, and this is coming from the University of Montana, which was a particle lab actually, and where we worked on inflammation and particles, as I looked down here at RML, and there was Katie's lab, and she had a lot of things on metabolism actually. That's kind of the direction she was going in, and this was so one of the things that the bug does is it alters the metabolism of the macrophage. So this is a really new idea actually. This hasn't come about since very recently, and that it can really shut down glycolysis, which I know not everybody knows what this stuff is, but it can change the metabolism of the cell to suit its needs and to create a niche or environment in which it can replicate. And how it does that is still, there's some things we do know, very simple, but there's a lot that we don't know. And so I came in, and that's the project I started on, is working on the metabolism of the host cell as well as the bacteria and how they interact to facilitate the pathogenesis of Francisella. So it's a very interesting project. And metabolism as well. I have a paper that I'd gotten in autophagy, and autophagy plays very much into metabolism. It's an integral part of metabolism in a cell. And so I saw this and I was like, that is really cool. And as all all senior graduate students should do if they're thinking of going into a lab, they need to read the papers. It doesn't look good if you don't. And so I've been reading through a lot of those papers, and I was very interested in what Katie was doing. And it just happened to be right down the road as well. And so I was like, well, that's kind of a win-win situation. I have 3 kids, and I was in graduate school, and it would have been difficult to move, and so seeing this right down the road, a perfect opportunity and a very good, well-renowned facility with great PIs and PIs as a principal investigator, mentors, resources. It was just a win-win situation. I don't think you could have asked for better, actually.
So what is your greatest science moment?
Oh, I've had a lot of the moments like that have been described, but I think probably the greatest one.
Not the goat scat, right?
No, those were really good, but honestly, I think I can pinpoint it down to maybe 2. One of them was getting funded.
Yeah, okay.
I got a fellowship, and I had applied to the NSF for the graduate program. I'd tried applying for that one, got like second place, what you might call a second place. It's the honorable mention, but no money. That doesn't.
No planet either.
Yeah, no planet.
Seems like everything's downhill after you
Yeah.
plan.
I applied for the Department of Defense grant, and this is a lot of time to put into these, and been turned down. And then I applied for a Pharma Foundation fellowship, and they give about 8 of these out a year nationwide, and my wife can testify to this. There's nights I'd be sitting there pretty late, typing these things up. Oh, delete that paragraph. That did not sound very well. And just over and over again. I swear you rewrote that grant like 4, 5, 6, 7, 8, 9 times. I don't know. It was over and over and over again. And to get a product that looked really good, and then you're comfortable looking at this grant, that it shows your logical thought process, and then you submit that knowing that you had done a good job, that it was a good grant whether it got funded or not, and then you get this, and then you're kind of waiting. It's like you get that really nervous turn in your stomach when you see the email of your reviews coming back, and pull it up, and it was, yeah, you've been funded. And that was a big relief. It was very nice and a very exciting time to get that. And it really helps on a resume to get that. And I think the other time was getting my first paper published too. It was very, very nice. But,
So what's in your future? What are you going to do?
I plan on going the whole 9 yards here. So once I get done as a postdoc, whether intramural or academia, I would like to be a principal investigator and do some high quality research in either of those areas. I haven't committed to either one of those because right now I think with the way funding is, you do have to be open to what comes your way. But that is the future of where I would like to go.
All right. Marie, what do you think is the single best way to get teenagers interested in science?
Well, I really do think that when they see the relevance in their daily life, that sounds kind of cliche, but I do think that makes a big, big difference. I've been so happy teaching health enhancement because it is the easiest sell ever, because every kid knows that they should be healthy, they feel good, they're happy. And then I have that avenue to teach all kinds of things to them, like infectious disease and the immune system and that kind of thing. And as I was listening here to Forrest talking about tularemia, I thought, I understand that. That's because Sam, the other day in class, taught his class about tularemia. And he did this very fun PowerPoint with a weird little guy who kept popping up all the time. And everyone was like, what's that little guy? There was all this humor. But he was able to explain some very complex things in a comprehensible way. His classmates are kids all the way from special ed to, the National Merit Scholars. And he was able to just explain that. And those are the kind of moments as a teacher that, I just hold on to and try to think back to when I'm having a bad day, especially, because it's awesome just, again, to see those kids being able to process information, put it in a relevant form for themselves and explain it to others.
Why did you become a science teacher?
Well, kind of like Jim, sometimes it's just chance. I have a degree from the University of Washington in zoology. My idea was I was going to be a game warden. So I graduated, came back to Montana and put in my application, and they're like, we hire a game warden like once every 10 years. And we just hired one, so, So one of my brothers got married in Bozeman, and I was out with my 7 brothers and sisters. We were having a good time afterwards, and there was this really cute guy there, and I asked him to dance, and he said, sure. And so as we got to know each other, I found out he was a burn nurse, but he didn't want to do that anymore. He wanted to be a science teacher. So I thought, wow, maybe that's what I should do. So I decided to go back to school, and because we thought, wow, that'd be a lot of fun. And so we did. And when you were talking about your friend David Quammen, when I was showing him Montana and teaching him what a great place we had to live here, we would read David Quammen's Natural Acts to each other in the car as we were going down the road. So it was just something that sort of fell into my lap, and then I found out that I loved it.
There was no point when you wanted to be a scientist, was there, like Jim maybe?
No, I wanted to be outside with animals. And so now I'm inside with animals. And sometimes we go outside.
So even after so many years of teaching science, you don't want to be a scientist, right?
No, I really don't.
Okay.
I just absolutely love what I'm doing. And when I go down to Walgreens and I see one of my former students there, and she's checking me out, and she's, oh yeah, Miss Antonoli, we were just talking about some stuff yesterday, and remember when we were doing that when you're talking to us about whatever it might be. Those are just, in a small town, these are the kinds of things that we just cherish, that we have such a tight community here, and so no, I don't want to be a scientist.
So do you have I asked everyone else about a great science moment. Do you have a great teaching moment?
Oh, there's just too many to even mention. I just, Recently, my husband and I both coach extracurricular science competitions, and we've been doing it for over 25 years, and we've always done well. When Jim was at Corvallis, we would always go, neck to neck with Corvallis. But in the last 2 years, we've won the state competition, both in Envirothon and Science Olympiad. So after all those years, we finally made it that far, and that's been a joy. That's been really fun to have that happen.
What's in your future?
Well, I think I'm going to be here a good long time. I think that I like what I'm doing. I'm excited. There's so much variety, and so I think I'll be teaching for a good long time.
You'll be here teaching science and doing all these things with RML that you've been doing.
Yeah, I just hope to continue that partnership. And, science is a team sport, so to speak, and not just Rocky Mountain Lab, but all kinds of people, science professionals in our community have been on our team and pitching in to help us with all kinds of things, being mentors for our Science Olympiad and our Envirothon, and helping us to devise units, and guest speakers on and on and on. So yeah, I hope to continue that relationship and make it even stronger.
Yeah. So Jim, how do you get teenagers excited about science? What's the best way?
Just get excited yourself.
Ah.
Yeah. I think that's what always worked for me. I could see the kids excited. And I had a mentor in high school, like you said you did, that inspired me. But I remember a favorite teaching moment. One time I had designed a lesson in stoichiometry in chemistry class. And stoichiometry doesn't come easy to most kids in high school. Most of them can't even say stoichiometry. But I had designed the lesson, and I had to design a lesson from scratch for it was some teacher competition or something. And I just remember after I had made the lesson, I didn't care about the competition, but I just watched how excited they got, because I was so excited about it. And every one of those kids got it. They understood how to use the equation to figure out how much glucose they had burned, blah, blah, blah. It was just I just remember the feeling of, that great moment. But to me, that's how I got kids excited usually about science.
Do you have a great science moment yet, or is it too early?
No, I've been there for 10 years. I hope I have one. Well, one of the bummers about studying prions is that prion diseases take a long time, right? They have a really long incubation period. So when these virologists do an experiment that's 7 days or 10 days and they got an answer, but for prions it's 150 or 300 days. So my science moment was in a prion experiment, the first big one that I had that I was in charge of. We were testing a human mutation in the prion gene, but it was in a transgenic mouse model. So the mouse was expressing the human a prion protein with the human mutation. And 150 days we got this answer, and it was great, but then I had to wait another 150 days to check it. So we did that, and we got the same answer. So that was 300 days, that was a year. And then we decided, well, we should try these other strains because this will prove that that's right. So at the end of like 600 days, we got the answer, and I was really excited.
You have to be patient in prion field, right?
Yeah, you do.
So what's in your future?
I think I'll keep doing science at Rocky Mountain Lab. It's a pretty special place to do science, I'm so close to the wilderness, which is important to me, and a small community. I think it's a pretty special place to do science.
I guess you'll continue to work with the high school and people like Marie and others and help bring science there as well, right?
Right.
Because you have that connection.
Yes.
So don't forget it, right?
I won't forget it. We have a community college here too, and I don't know if it's common knowledge or whatever, but a lot of postdocs from Rocky Mountain Lab teach at the community college. So that's another really nice way that we've been able to get high quality teaching and get the college really rolling.
Well, as you see clearly, having big science in a small town can lead to science careers, and this is very impressive. You should be proud of these individuals and what they do, You know, in New York City, we have lots of science. Who knows what impact the science has on high school? It's just too diluted. We can't tell, but this is a great experiment right here, because you can see things happening, whereas many other places you can't. Now, this has been an episode of TWiM, This Week in Microbiology. We record this every other week. You can find all the episodes at iTunes at microbe.tv/TWiM, or you can find it if you have an app on your cell phone for getting podcasts. You can get it there as well. Within the next few years, every car sold in the U.S. will have the podcast app on its dashboard, and you'll be able to play them there. That'll be the end of radio and the beginning of podcasts forever, so you'll be able to listen to them there. Consider becoming a patron of TWiM and all the other Microbe TV family of science shows. Go to microbe.tv/contribute. There are many ways you can help us do our shows and do more. You can subscribe for as little as $1 a month if you'd like, or there are other ways. On this show and many others, we answer questions and comments during our normal episodes. If you want to send some in, you can do that at twim@microbe.tv. I want to thank all the participants today for joining me from the University of Montana. Kyle Shiflett, thank you so much. Hope you enjoyed it. From Rocky Mountain Labs, Brian Hansen, thank you.
Thank you.
Rocky Mountain Labs, Forrest Jessup, thank you very much.
Thank you.
Enjoy that trip to that planet. From Hamilton High School, Marie Antonioli, thank you so much.
Pleasure.
And for everything that you have done as well. And from Rocky Mountain Laboratories, Jim Strebel, thank you so much.
Thank you.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank Marshall Bloom for organizing my visit. Ken Pico also. Where's Ken? Is he around? Thank you, Ken, wherever you are. He's gone. For doing lots of... Thank you, Ken. Thanks for doing publicity and background on the applicants. He made me look smart here today. I also want to thank Rocky Mountain Laboratories for supporting my visit. Thanks to the audience for coming as well. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology. Thanks, everybody, and good night.